See everyone. Fantastic. It's nice to see everyone. Thank you for joining the June SFPM seminar. It's a really exciting time for the organization, and I think as you'll hear today, today's talk is going to be nothing less than extraordinary. I'll turn it over to Keith Ligon in 1 minute to introduce today's speaker, but I wanted to give a couple of updates off the top. As everyone knows, our colleague and board member Jeff Tyner, together with Keith Flaherty and Tony Latai and others, are organizing a workshop that everyone is invited to. I'll put again the link in the chat. This is coming up on July 1st and 2nd at the NCI to think about a functional precision medicine future for the United States and beyond. And so Tony, of course, as well as Jeff and Keith, encourage everyone to participate. There's a remote option, so SFPM would strongly encourage you to sign up. And the SFPM is prepared to host some additional listening sessions and learning sessions after the workshop to make sure that the members feel included in the outcomes and that their voices are heard. So to kick that off at next month's seminar in July, we're going to have a special session where Jeff and Keith, the two leaders of the workshop, will talk with SFPM members about how the workshop went and what were some of the key learnings. And you'll have a chance to ask them direct questions and provide direct input. And we expect to have additional listening and learning sessions as the summer and fall go on. The SFPM is actively fundraising and building biopharma partners. You'll hear more about that in the future. And there are a number of organizations which are contributing funds to help support the cause. Those funds are really important because they help us to convene, host events, and soon begin to build flagship projects that will show what the SFPM is capable of. That's a big reason why your membership dues are so valuable. So we continue to increase the number of members in good standing. If you haven't paid your $100 donation, I'd really encourage you to do so. That's very helpful. It allows us to continue to serve you at a very high level, and it really increases our ability to recruit biopharma funders because we can comment on how many paid members we have. So Aaron, maybe you could put the link again in the chat. I'm hopeful that anybody that hasn't signed up but is participating in these webinars might pay their annual membership due during this call. And there's an annual subscription option, just like Netflix. So you can just click on that and you'll get charged as the year go on. Hopefully you're getting more than $100 of value from the organization. And if you're not, I want to hear about it because we want to increase the value. With that being said, are there any other announcements that I forgot, Alice, that you wanted to make, or Keith or Aaron, you wanted to make before we introduce today's speaker? Alice, I think you're muted if you're trying to make some comments.
I still am. Sorry about that. I just want to mention that we heard from many of you that the kind of membership platform we had was a little bit clunky. So the link you see here is our new system. Should make it very easy for all of you to, pay your dues. So if you have any trouble, please reach out to us. But hopefully this is simplifying the process. And so, yeah.
And I should have mentioned, we're in the middle of strategic planning. As I mentioned in previous calls, we've recruited a nonprofit consultant that's helping us, and all members that are in good standing are receiving a survey at which you can provide input to drive the strategic planning and branding and mission statements for the future. So if you haven't returned that survey, please do. If you haven't received the survey, please pay your membership dues and you'll receive it. And Alice and I and the rest of the board would really appreciate your feedback.
Yeah, we're going to recirculate it again for those of you who didn't respond. And, your opinion on the future of the field matters, certainly matters to us, and it's going to help us shape the vision moving forward. So please take a couple of minutes to answer the survey.
Any additional announcements? Joanne, Keith, Aaron, anybody else, any other board members? All right, fantastic. Keith, let me turn it over to you to introduce today's speaker.
Yeah, it's a delight to have Sri Raghavan joining us to share some of his exciting work on modeling. And just as background, Sri is an assistant professor at Harvard Medical School and an independent investigator in the Department of Medical Oncology at Dana-Farber Cancer Institute, where he's a medical oncologist in our GI Cancer Center. He's also an associate member at the Broad Institute at MIT, which is, I think, some of the collaborations he's going to talk about today, which are very exciting. One in particular is this Project Ex Vivo, which is a collaboration between his group and several people at the Broad, and also a Microsoft research group with kind of a focus on getting AI sort of leveraged for increasing understanding of drug discovery. And as its title implies, cell states and plasticity, and how that's new knowledge that we're understanding lately. And certainly appreciate your prior publication, Shri, which really did give us a better nuanced understanding, some about the good and bad of some of the models. So we look forward to your talk, and I'll turn it over to you. Thanks.
Great. Thanks for the kind introduction. I'm happy to be here and present some of our group's work on defining, modeling, and targeting cell states in cancer. These are my disclosures. And so I'll talk a little bit about our prior work and then sort of where we're headed in the currently and in the future. If people have questions along the way, I'm happy to sort of take questions during. I always think it's nice to have it be a discussion rather than just saving everything for the end. So feel free to interrupt if you can, if you have a question. So when we think about precision medicine, I would say that by and large, DNA mutations are the cornerstone of how we've begun to stratify and treat patients over the last many years, where through advances in genomic sequencing technology, we've been able to identify patient cancer-specific alterations. A key step here is being able to effectively model this in a variety of systems, cell lines, organoids, mouse models, and this provides the substrate for drug discovery, mechanistic understanding, et cetera. And then this gets translated into stratifying patients and therapeutic selection. And so this pipeline has worked very well if the major feature relevant to drug response is mutationally encoded. And for this group, I don't need to go through. There are numerous successes over the last 20 years or so. Most recently, excitingly, as a GI medical oncologist in the pancreas cancer targeted therapies. But there are still some challenges remaining where many patients still lack clear, actionable alterations that have drugs available. And then even for those patients who do have targeted options, most of them develop resistance either due to cellular heterogeneity or adaptive resistance through both genetic and non-genetic mechanisms. And so our team was really interested in trying to understand what other cellular features influence therapeutic response and resistance. And so as Keith and Jesse alluded to, we think a lot about cancer cell state. And so, when you think about cancer cells, there's the underlying DNA sequence, the mutated blueprint, but this is then dynamically transcribed and translated into RNA and protein expression that can lead to a variety of functional states. And a variety of inputs determine cell state, including, of course, the underlying alterations, DNA alterations, but also epigenetic states, interactions with the tumor environment. And cells integrate all of these different signals into sort of functional outputs. And so our team really tries to understand, how do you begin to define cell state as the groups of genes co-expressed by a cell that captures both cell function and environmental context. And so, as I mentioned, many of our drug pipelines are largely aimed at the DNA alteration level, in part because we have great tools for both measuring and studying DNA alterations. How do we begin to extend this to understanding RNA expression phenotypes and associated functions? And can we, over time, begin to develop cell state based, using cell state as a biomarker for therapeutic selection? It's sort of a big question that we're trying to ask. And so our team has sort of three major pillars that we're focusing on, all geared towards trying to relate cell state diversity to functional targetable attributes. And so one major effort is trying to benchmark and engineer cellular models that better reflect cell states, as in our case, measured by RNA expression that you observe in patients that have functional consequences. How do we effectively model these in cell line organoid systems? In the second part of the talk, I'll really focus on trying to build systematic tools for understanding context-specific functions of cell state. And then lastly, I won't spend much time talking about this, but this is the crux of our work with Microsoft Research, is how do we begin to use larger scale computational models to infer state behavior liabilities with these sort of predictive foundation models, and what types of data need to go into these models to sort of essentially boost their predictive power. And so focusing on this first pillar, if you will, I think one of the things we learned through my postdoc and early faculty career is really the question we were trying to ask is, do current models represent cancers well enough to map cell state dependencies? And really, what clinically relevant features do we effectively model, and are there ones that we're missing? And this project started out, my lab focuses on pancreas cancer and also in biliary tract cancer, a subset of liver cancers. And so focusing in pancreas cancer, where we've known about the tumor genetics for decades, there have been, until now, few genotype-directed or positive immunotherapy studies, although obviously now changing with the advent of the RAS inhibitors, in particular daraxonrasib for pancreas cancer. Importantly, tumor cell state has been shown through multiple bulk RNA sequencing studies where cancer cells fall into one of two major categories by bulk RNA sequencing, a basal-like phenotype and a classical phenotype. And these are strongly associated with overall survival, where patients with more classical phenotypes, and admittedly, this is in a cancer where most people do fairly poorly, at least hopefully the RAS inhibitors will change that. But even within that subset of patients, some patients, those with more classical expressing tumors, tended to do better, have better upfront chemo responsiveness, and overall better survival compared to patients with more basal predominant tumors. And so with this as background, our team, in collaboration with Jesse, with Brian Wolpin, Andy Aguirre, colleagues at the Dana-Farber, and our whole clinical research team, we sort of, I guess now 10 years ago, started a large effort to collect patient samples, generate patient-derived organoid models with this idea that, oh, if we derive a sample from a patient and then do sort of real-time drug testing, we should be able to predict responses and maybe be able to select options for second-line or third-line therapy. And so we did this. We undertook this large effort where we collected a number of patient samples. I mean, I think now we have between my lab and Andy's lab and the Broad, I guess probably 100+ organoid models from pancreas cancer. But we found that despite doing this drug testing, we were actually surprisingly poor at predicting, at sort of correlating drug responses in the organoid models with those in terms of patient outcomes. And so we wanted to take a step back and try to understand, are there features that we're not well reflecting in the organoid models that might be contributing to issues with sort of the correlation between in vivo and in vitro. And so we first looked at sort of the canonical driver alterations in pancreas cancer across a cohort of primary and metastatic patient samples, TCGA plus a metastatic cohort primarily of liver biopsies, and then compared to established two-dimensional pancreas cancer cell lines and our organoid cohort. And at the genomic level, the frequencies of these driver alterations is basically equivalent across these different contexts. But interestingly, when we looked at sort of the RNA subtypes, we see major model-specific skews. And so specifically in patient samples, where every column here represents an individual patient, the patients span a spectrum of classical to basal, both in the primary and the metastatic settings. But interestingly, nearly all of our established cancer cell line models are basal in nature, and nearly all of our organoid models that we generated were classical in nature. And so this really highlighted that cell state representation is skewed by model type, suggesting a heavy influence of the growth environment. And so we wanted to investigate this further, especially given the clinical data that RNA subtype strongly correlates with chemo responsiveness and clinical outcome. So basically, if our models are so skewed, are we effectively able to represent what you might see in patients? And so to do this, we developed a workflow where we compared in vivo and matched model phenotypes, where we took a cohort of metastatic pancreas cancer biopsies, predominantly the liver. We dissociated them, and then we performed single-cell RNA sequencing on this patient biopsy using the seqWell method developed by Alex Shalek's lab at MIT and We work closely with Alex's group. Peter Winter and Andrew Navia were close collaborators, postdoc and graduate student in Alex's lab at that time. And importantly, in parallel from the same sort of single cell suspension, we generated patient-derived organoid models and serially passaged these and also assessed them by single cell RNA sequencing over time. And so this pipeline really allows us to directly compare in vivo to ex vivo phenotypes and to really dissect which features are preserved, which features are gained uniquely in the culture conditions, and which features that are seen in vivo are lost. Many of those typically seem to be associated with the TME signals or due to selection of specific subclones in vitro. And so, I think one of the things we sort of learned about and thought through this process that making a model is essentially an experiment. And you're sort of testing what are the things that are happening or changing as you go through this process. I see there's some stuff in the chat, so let me just pull this up also on my side. So,
It's okay, Sri. We'll call out questions for you. Keep going.
Great, thanks. Great. So using this pipeline, this is sort of our cohort of patient samples. So the in vivo cells where we scored every cell. Columns are single cells. Rows are gene expression for basal and classical programs. And as we saw with bulk RNA-seq, we see that there's this spectrum of basal to classical across this single cell cohort. Single cell resolution did allow us to see that there's a population of cells that co-express both phenotypes. And we sort of enriched for a program that is unique to this intermediate co-expressor subset. A different way of plotting this, which I'll show in subsequent slides, is this what we call a cell state diagram, where the x-axis is the basal to classical sort of commitment score. It's basically the classical score minus the basal score, and the y-axis represents intermediate co-expressor expression. And so every point here is an individual cell scored for basal, classical, and IC, and it sort of forms this tripartite distribution when you look at cells emerging from patient samples. In contrast, when you look at sort of the cells from early passages of matched organoids, and this is pretty early, like passage 2, what you see is a dramatic skew towards classical expression in the organoids. And we were surprised by how quickly this occurs within 2 to 3 passages out of the body. So really a couple of weeks by the time this happens. And so when you see this type of skew, again, consistent with what we had seen at the bulk level, we wanted to ask, can in vivo state representation be recovered in our model systems? And so to do this, we established a pipeline, which I'm not going to go into great detail here, but essentially we nominated autocrine and paracrine signals from our in vivo cohort from the single cell data from patient samples. We then compared those to our organoid culture system and then re-added back in state-specific factors that we thought might influence the expression subtype. And so this is just one such example. We did this for a number of other cytokines, including interferon gamma. But in this case, we took this organoid model, heavily skewed towards classical. And as you add in TGF-beta, over time, you can see that the distribution shifts towards a more intermediate IC and basal expression pattern. I'm not showing this here, but if you withdraw the TGF-beta, the models shift back to classical, really highlighting that 2 things. One, that TME signals are critical in terms of determining some of these cell state phenotypes. And 2, these models are, many of these models are quite plastic and responsive to signals in their environment. And, I think one thing that, we sort of an interesting anecdote is that it turns out standard organoid culture actually has a TGF-beta inhibitor in part to limit fibroblast outgrowth. And so, I think one reason why why you're pushing cells towards the classical phenotype is you have an inhibitor in there, they can never sort of receive that signal. And so we had to take that out in addition to adding exogenous TGF-beta. So I think just highlighting that the things that we put into our media, I think historically the effort has been how do we get a model to grow? And I think not always how do we match the phenotype to maybe a relevant phenotype in patients. So now, I think we wanted to get back to, does this ability to tune models, does this have functional consequences? And so to do this, we performed an experiment where we now took a series of patient-derived organoid models and then sort of isogenic pairs that were tuned to either be more classical or more basal, in this case by manipulating their media conditions. And we first tested their responsiveness to standard of care chemotherapy used in pancreas cancer, gemcitabine, paclitaxel, and SN38, which is the active compound for irinotecan. And what we observed is that the classical models were more sensitive to these standard of care chemotherapy agents. And when you look at clinical data, and so this is data from a study led by Grania O'Kane and Jennifer Knox from the Toronto pancreas cancer group. They took upfront metastatic patients, subtyped them with RNA-seq and a GATA6-ish assay. And they observed how these patients responded to upfront chemotherapy, where you can see that patients with classical enriched tumors tended to respond better. The y-axis is sort of tumor size change. It responded better to upfront chemotherapy, whereas the patients with more basal predominant tumors tended to respond less well to upfront chemotherapy. And so this pattern kind of mirrors what we're seeing in these isogenic organoids that have been tuned now, where the more classical models seem to be more sensitive to chemotherapy. And so, gives us some in vitro evidence that cell state can directly impact drug response. So we extended this drug panel to a larger cohort of agents. And these are a little bit older, but several of these were selected in part because they were available sort of through various clinical trials in our phase 1 group. And then others were sort of tool compounds for interesting pathways that we thought might be interesting. And so what was notable here is that while the classical models seem to be more sensitive in general to chemo and DNA damage repair agents, the basal models tended to be much more sensitive to MAP kinase-directed inhibitors, BVD523, which is an ERK inhibitor, trametinib, a MEK inhibitor. And interestingly, data subsequent to this has also shown that the basal models tend to be more sensitive to upfront RAS inhibition. And so, this effort really highlighted, one, that cell state impacts drug response, suggests that maybe we can begin to perform systematic screening for state-specific therapeutic options. And so, as I mentioned, more recent data through our group, from Andy Aguirre's group and others across the country, have highlighted that RAS inhibitors also seem to preferentially target cells that are more basal expressing. And there's some ongoing efforts to try to understand why that is mechanistically. We were also interested in, okay, if classical cells, are there targeted options that might work in the classical context that could potentially be useful as combination therapies? And I'll just add that now with the daraxonrasib trial results for second line, there are now trials underway for testing upfront chemotherapy plus RAS inhibition in the first line with some promising results. And so that data is still will be coming, I think, over the next, hopefully 6 months to a year. But already people are thinking about, combinations with chemotherapy, and we wanted to see, are there other combinations with other targeted agents that might work? I'll also state that I didn't show this data, but when you look across a cohort of pancreas cancer patient biopsies, actually most patients have populations of cells that are basal and populations of cells that are classical. It's not like every tumor is all classical or all basal. So there's heterogeneity within a patient that probably is important to figure out how to target from a drug standpoint. And so for this next effort, we really started looking at a large effort that was started in part by Jesse, along with the Broad Cancer Dependency Map program, Paquita Vasquez, Sukasa Shibue was a research scientist who is in DepMap and is actually now part of our team. And then samples donated, provided from many collaborators, including Keith, who provided a number of the CNS brain neurosphere models. Andy Aguirre, myself, we submitted a lot of our pancreas samples. Adam Bass, who submitted a lot of gastric and esophageal samples, among many others across the country. And so the DepMap called this the
Hey, Srini, but before you move on, Sanam had one question about TGF beta and MAP kinase.
Oh, yeah.
So Nam, I think you're asking if the MAP kinase results could be due to TGF beta activation in the basal setting. Is that correct?
Yeah, exactly.
Yeah,
Just
that's a great question. So that's a good question. We don't, I don't have the answer to that specifically. That's actually one of the sort of sub aims of my KO8 award that we're in the process of trying to investigate, dissecting sort of canonical versus non-canonical TGF beta
yeah.
activation versus the MAPK. But yeah, totally relevant question, and hopefully we'll have an answer for that in the coming year or two.
Thank you.
Yeah, so here, this is a large compendium of models, organoids, neurospheres that have undergone whole genome sequencing, RNA sequencing, bulk RNA sequencing, and then genome scale CRISPR loss of function screens. And so, we became interested in this and trying to understand what can be learned from cross-lineage analysis of next-gen models. And part of this was driven by the observation that, as I had alluded to earlier, we see classical expression enriched in pancreas organoid models. It turns out this is actually a phenomenon observed across multiple GI lineages, where on the left is enrichments in organoid models versus in 2D cell lines, and what we're calling the PDAC classical, just because it's sort of related to the classical signature, but these are signatures from a Taroche 3CA cell expression state paper. Sort of the PDAC classical is the most enriched in the organoid models. And on the right, we sort of Sukasa plotted these against sort of TCGA. So the circles represent TCGA samples and their distribution along a classical score axis compared to the 2D cell lines, the traditional models in green, which are skewed towards being less classical, more basal, versus the organoid models, which are overlap the TCGA distribution more sort of completely, because they tend to be more classical. And we see this in pancreas. We saw this in gastric and esophageal cancers, and then also in colorectal. And so with this sort of representation of classical models, which are not really present in the 2D cell line space, we asked, are there dependencies associated with this cell state? And so what's striking here is when you look at in the organoid systems, classical associated dependencies, it's really enriched for Wnt pathway dependencies. So MESD and Wintless, and the pathway is shown on the right here. These are important for sort of secretion of Wnt and sort of stabilization of some of the receptors. TCF7L2 is the downstream transcriptional effector. Frizzled5 is a co-receptor for the Wnt ligand. It's just striking how many are enriched in this classical subtype. And so is there a potential opportunity for therapeutic development? And I'm not showing this here, but the obvious question is, okay, well, if organoid media has Wnt in it, is that just why you're seeing this Wnt dependency? It turns out many of these models that were screened actually did not have a Wnt-rich media. And so we don't think that exogenous Wnt is a confounder here. And we've been doing some work to try to confirm this, and that has held up so far. And so, I think the question here now that we're trying to address is, can we use these data, these models now that represent classical, that represent basal, to more effectively identify classical specific targeting agents such as the Wnt therapeutics that might complement things like the RAS inhibitor for the basal targeting side of things. And so current work, we think that this is an autocrine mechanism where sort of cells are secreting Wnt and sort of receiving it in the same cell. We're trying to dissect this more closely. Is this, I think one of the key questions here from a therapeutic standpoint, is this unique to cancers, particularly this autocrine mechanism, and is there a therapeutic window to target this? And, I think several years ago, companies had tried therapeutics, particularly around the porcupine inhibitors for targeting the Wnt pathway, but many of those studies were not particularly positive. But I think looking back, those studies were largely not selected. There was some selection for RNF43 mutant or are spondin fusion patients, and those patients tended to show greater responses to those porcupine inhibitors. But many of the patients were sort of unselected. And so one of the hypotheses here is if does sort of a classical cell state or some subset of that provide an additional biomarker that might select for patients who are inherently more sensitive to Wnt pathway suppression. And interestingly, in recent literature, paper I think earlier this year or last year, I think it's earlier this year, in colorectal cancer, suggests that Wnt pathway activation may be one route to RAS inhibitor resistance. Again, highlighting that maybe sort of this type of combination strategy leveraging sort of the cell state association might provide a new combo option. And so, our team is really trying to understand and sort of pursue this further, not just in the Wnt side, but are there other routes for defining state-associated therapeutic targets, and really highlighting that we think that updated benchmarked model systems will enable this effort. I think the one area I'll just comment here that we haven't really gotten to, but I think ties back into some of the maybe the functional precision medicine aspects is, historically, when we generated our organoid models, we just use one standard culture condition. And, one of the questions we've wanted to ask, but haven't had time to really go after is, should people be initiating models in multiple conditions to try to preserve the heterogeneity seen in a patient tumor? And then when you do functional testing there, does that help you maybe better predict what might happen in a patient? So I think that's an interesting question, translational question that we haven't really gotten to, but would be interesting to explore. Okay, so in the sort of next second portion of the talk, I wanted to focus on sort of now our efforts to sort of systematically map and understand context-specific functions of cell state. How do we systematically annotate relevant cellular programs, relate them back to atlas-type data, and how do we really ascribe function to different cancer cell states? And so, part of this is built on this idea that foundation models and even, many of our analytical tools are built on having the right references to tie a given biological feature to a function. So when we think about DNA alterations, we've built normal variant reference maps that allow us to identify, recurrently altered versus not, and then cancer-specific alterations that then can point to sort of functional consequences for a given genetic element. Similarly, for protein sequence and structure, we've through crystal structure efforts and now through some of the AI efforts, we can begin to relate structure and make predictions around function of given protein sequences. But when we think about a more heterogeneous biomarker like RNA expression cell state, everyone runs gene set enrichment analysis, maybe relates to some single cell atlas, but what's missing here is what's the functional consequence of a given state? Is it just an observation, or is it actually relevant to some functional attribute of a cell? And how do you begin to build out a map that ties cell state to function? And so to this end, we've sort of built a strategy that we're testing right now, and we're definitely open to feedback and other thoughts around this. So we call this sort of visualization the hypercube, where every plane represents an individual model, a cell line organoid model, and where previously we used culture conditions to tune cell state expression, we're trying to that is limited somewhat in scale. And so here we're testing whether we can use genetic tools, ORF libraries or CRISPR-I, CRISPR-A libraries to engineer transcriptional heterogeneity across a given model, and then look at how that heterogeneity confers selective advantage for those engineered cells to a variety of therapeutic agents or environments that would be typically not supportive of cell growth. And the idea is, can we do this across enough models? How much of the space do you need to sample to be able to understand sort of the rules for which cell states govern specific functions? And ultimately, how do you relate this now to interpret all the cell atlas type data coming from patient cohorts? And so the way we operationalize this from a practical standpoint is right now we're using a transcription factor ORF library. It consists of about 3,500 transcription factor isoforms. This was developed by Julia Zheng when she was a graduate student in Feng Zhang's lab. And so these are barcoded, can be introduced into a cell line or organoid, and then we perform positive selection screens where we're either testing cells against control, DMSO, or a selective pressure. We culture these over a period of time and then can extract the DNA and sequence these to see what's enriched under the selective pressure, which transcription factors are enriched under selective pressure versus the control. And so we chose a couple of different avenues, which I'll talk about briefly here. One is we focused on resistance to daraxonrasib, specifically using RMC-7977, which is the tool compound for daraxonrasib. And I think this was driven by partially my interest in clinical focus in pancreas cancer, where we see dramatic responses to this drug, but nearly all patients progress with some form of resistance after about 13 months, 9 to 13 months. And importantly, in sort of data from G12C inhibition across multiple disease types, what you see is that while about half, probably a little bit more, of patients have a clear genetic alteration that might be a putative cause for resistance, sort of downstream in the MAP kinase pathway, et cetera, there's a good 25 to 50% of patients who lack a clear genetic mechanism. And so with those patients, we're kind of stuck with what do you do next in terms of a therapeutic standpoint. And so we are also hoping that this approach of defining sort of non-genetic mechanisms of resistance may inform options for some of these patients where there's not a clear genetic driver of resistance. And so we did this for a number of models now where we looked at RAS inhibition. How do these models become resistant? And so this is just one example of a pancreas cancer cell line treated with the drug, GFP control versus the transcription factor library. And over time, you see emergence of clones transduced with the transcription factor library. We then collect these cells and then sequence the barcodes and count TF frequencies in the sort of resistant population relative to the control. And so this is just an example. Well, this is sort of data from our initial cohort of 3 pancreas lines, a lung line, and a colorectal cancer cell line, all with RAS alteration. And this is sort of cumulative hit calling where a number of these transcription factors score across multiple lineages. Others are more lineage specific, and many of these are represented within TCGA cohorts in terms of just average expression. And so we wanted to sort of do some validation of this, and we've done this in a couple of different ways. So we first built a mini pool of the top transcription factor hits, about 20 or so, and performed sort of a similar experiment, both using cell counting and barcode representation. Many of the top hits validate. Again, HNF 1 beta, 1 alpha sort of consistently seem to be among the strongest drivers. YAP1, which has been shown in other reports to drive RAS inhibitor resistance, is among sort of scores across multiple lineages. And then sort of some, as I mentioned earlier, some of the transcription factors are more lineage specific. And so we wanted to then begin to dissect, okay, how do we understand what's happening? What cell states are these driving? How do we understand what cell states are emerging by these transcription factors, and how do you relate that? What's the mechanism by which this is now leading to drug resistance? And so, sorry, I saw a chat message. We perform single cell RNA sequencing across a number of these engineered models and also the parental controls. And you can begin to sort of see sort of clusters of program similarity emerging both across lineages. And so we wanted to really focus in on HNF 1 alpha 1 beta, I think just from a bandwidth standpoint, as the strongest hits. And those are also known in the known sort of lineage-associated factors in the GI space. So we spent most of our time downstream of this focusing on HNF 1 beta and 1 alpha, as I'll show you. But there's a number of hits that we're trying to sort of further investigate. And I think if folks in the audience have something catches your eye, please let us know. We've not been comprehensive for sort of all the downstream stuff in terms of how to validate. So again, confirming HNF1 alpha, 1 beta resistance in now a number of pancreas and lung and colon lines. We again see robust resistance to RAS inhibition by cell counting. Interestingly, we see that these transcription factors induce cross-resistance to other MAP kinase pathway inhibitors, so trametinib and BVD523. And we've started to do some in vivo resistance experiments using cell line-based sub-Q xenografts, again, just confirming that HNF1 beta drives resistance in vivo in addition to in vitro in the presence of the RAS inhibitor. And so, the obvious question is this just happening because of ERK reactivation? That the transcription factor is somehow driving ERK reactivation. So we tested this using just sort of Western blotting, and I'm showing this in just one cell line, but we've now done this across a number of cell lines where in the presence of HNF1 alpha or 1 beta, you don't see phospho-ERK reactivation in contrast to sort of the parental GFP control. So we don't think this is just due to ERK reactivation. We're currently trying to figure out what is happening mechanistically. And so, we haven't figured this out entirely, but, this is sort of data now across a number of models analyzed with single cell RNA sequencing, GFP control minus drug in light blue versus plus drug in red. And then similarly for HNF1 alpha engineered models, HNF1 beta engineered models. So what we see, as one might expect, is that a number of genes are downregulated by RAS inhibition in the GFP control. But interestingly, that downregulation is somewhat blunted in the HNF1 alpha models. And also interestingly, we see a unique set of genes that are upregulated specifically in the context of HNF1 alpha plus RAS inhibitor. And many of these are driving just through enrichment sort of pathways that have previously maybe been associated with RAS inhibitor or MAP kinase pathway inhibitor resistance, STK signaling, YAP-HIPPO signaling, et cetera. And so right now we're trying to sort of wrap our heads around this, trying to dissect, okay, is there a specific node here that one can point to, or is this sort of multifactorial, and how do you begin to think about targeting this in terms of overcoming this resistance mechanism? I think one other sort of mechanistically interesting point here is that we seem to see, and we've seen this across other contexts too, that the presence of the transcription factor alone doesn't turn on all of these genes. It's really the intersection of the transcriptional state and the therapeutic insult. And so we have some early ATAC-seq data that we've done that still sort of analyzing here, but trying to understand what's happening at the chromatin level, also downstream of sort of HNF plus the drug, plus the drug. I see there's a question here. Have you considered using pathway analysis tools to identify resistance mechanisms? Yeah, we haven't tried this Hypatia tool. Certainly a useful suggestion for us to test. We've sort of done sort of generic sort of enrichment type stuff and are in the process of sort of testing some of the pathways that we've nominated. But I haven't tested any of the more specific options. So certainly useful to try to take a look at that.
Shri, you started with the idea of states as kind of a macro concept, and now you're honing in on individual genes, master regulators that drive those states. Which do you want us to focus on? Is it the resistance genes, or is it the census of states and the manipulation of the states themselves?
Yeah, so that's great. So we think that, so we think that we focus on HNF1 alpha mainly because to try to have a handle on finding sort of, to try to, have a resistance mechanism at least. But really what we think are more important are the states underlying, and the transcription factors are really a tool for us to get a handle to engineer models that are representative of different states. And so one of the points that we're working on, I'll get to in a couple minutes, is just how do we, now that we have essentially state-defined isogenic models, can we begin to use those to test systematically for downstream vulnerabilities using CRISPR or drug screening?
Yeah, because I think that's a big question that all of us are struggling with. Many approaches to FPM many media formats, many
Yep.
metadata formats. How do you inventory states in a sophisticated way to credential results and compare them to states generally, regardless of what model system
Yep.
you're using? And maybe we can get to that at the end.
Yep. Yeah, no, that's something that we struggle with. I don't know that we have a definitive answer, but we're hoping that some of these state mapping approaches provide us sort of the landscape of maybe functionally important states that we can then use toward that end. I think one of the sort of caveats of in vitro transcription factor positive selection screens is that you may find hits that are in vitro specific. And so this is definitely sort of still work in progress. We can definitely see that some of the programs that we are driving with our transcription factor overexpression are expressed within our biopsy metastatic patient cohort. These were all samples that were collected pre-RAS inhibitor, but we're currently sort of collaborating with Andy Aguirre, Brian Wolpin, Jonathan Nowak at the Farber, among others, to sort of look at samples from some of these trials where we are collecting tissue, pre-treatment, and then on treatment, and ideally post-progression, also have matched blood samples serially over the course of treatment. And we're leveraging sort of spatial transcriptomics. And this is really work being led by Winston Becker, who's a postdoc medical oncology fellow in Andy Aguirre's lab, using sort of spatial transcriptomics to try to understand what's changing as you sort of move forward through therapy. And so this is just sort of early data. Most of the samples so far are pre and on treatment. Many patients just clinically, when they progress, they don't necessarily want to get a post-progression biopsy. So we're still trying to collect more of those. This is just showing that even with spatial transcriptomics, you can sort of see a drop in sort of MAPK signature expression with the drug. And so what we're really waiting for is if we have resistance samples, are any of our resistance programs enriched in that cohort? And I think that data will probably be expanding quickly over the next year, just because these drugs are likely to become, almost certainly going to become clinically approved, and there will be many more patients to collect from. And so to sort of the work that's ongoing in this space, and then I have one more brief vignette, and then we can maybe have a few minutes to discuss, is we're trying to sort of understand using attack-seq and cut and run to sort of understand chromatin level regulation, additional in vivo validation. And then, as I mentioned, sort of starting to get to, can you build a series of isogenic models that all represent different RAS resistant states that you can then do downstream CRISPR or drug screening? And are there going to be shared targets that might sort of leverage that state resistance as a way that you can then target in a sort of biomarker directed manner? So we're also interested in sort of trying to understand, okay, this is one therapeutic example. We're doing this for other therapies, EGFR inhibition in lung cancer, ROS and ALK, other targeted agents in lung cancer. We also wanted to ask what happens with interferon tolerance, for example, as a microenvironmental pressure, with the ultimate goal of really trying to build a fitness map to this point where you can now map out TFs and associated states that now are tied to a functional consequence. And I think this is where the smaller transcription factor library allows us to scale across perturbations. And so this is sort of similar data for interferon gamma across melanoma, pancreas lineages and lung cancer lineages. Again, some are common, some hits are common, some are more disease specific. We've done some validation in this space. And then what's notable here, again, we see different sort of clusters, PDX1, TP63, and 73, which are strongly associated with squamous phenotypes in pancreas and in lung cancer, enriched for interferon tolerance. And so we're trying to examine this mechanistically. How are these transcription factor programs are seen in patients driving potentially immune mediated tolerance? And what's notable here is that we went back and sort of looked at enrichment of the signatures that we're deriving from in vitro overexpressing TFs against TCGA samples, and many of these actually portend poor overall survival in sort of in a TCGA cohort. And so this is also where we're trying to sort of I think it's important to really relate what we're seeing in the laboratory to what's happening in the clinical setting. So this is just another example of this. We had one patient, pancreas cancer, pre and post-treatment biopsies with an immunotherapy trial in between of a PD-1 and a macrophage targeting agent. And in this case, the biopsy was taken from the same liver lesion. And we can see that some of these transcription factor derived programs that we're identifying in vitro are enriched in the resistance sample, suggesting that maybe this P63 derived program is in fact representing an immune tolerant program that can be seen in vivo. So again, N of one example here, we're trying to sort of build out our cohorts. Unfortunately, in pancreas cancer, most immunotherapy doesn't work. So there's not a lot of tissue samples here, but obviously in lung cancer and other tissue sites where these agents are used more commonly, we're trying to sort of compile some of these data sets to try to, again, relate the states that we're seeing in vitro as resistant to interferon exposure, for example, are they reflected in patients? And again, as I showed in the sort of bar plot earlier, but when you go back and look, several of these transcription factor driven programs associate with poor survival across a number of cancer lineages in TCGA, whereas interferon gamma response signature is really only highlighted in melanoma. And then we've gone back, TCGA now has a number of sort of immune metrics associated with these. Some of these seem to be, some of the places where we see this increased association are seen in context with greater immune infiltration. So now we're trying to go back to the lab and sort of mechanistically test this in sort of some co-culture assays and that sort of thing. And so just so we have enough time maybe for a few minutes of discussion and questions, I just wanted to highlight sort of the goal that we're trying to build towards is how do we begin to build these cross-tissue cell state fitness maps? When we begin to understand conserved drivers, conserved states across tissue contexts, and ultimately now, how do we build this set of tools, isogenic paired models that can be used for downstream drug discovery? And I think the other piece of this is how do you now associate all of this with the large number of sort of HCA, Human Cell Atlas type data sets, and now begin to contextualize for hopefully what we're seeing in those large data sets. And so this is sort of ongoing work around ways to generate heterogeneity, other ways to generate heterogeneity, more comprehensive mapping, trying to understand how plastic are these resistant states. And then lastly, through this collaboration with Microsoft, we're trying to sort of understand a number of questions around this, like how much of this type of data do we need for more complex foundation models to be able to predict some of these state-associated therapeutics or resistance mechanisms? What do we actually want any of these models to be able to predict? And then I think on the more basic science side, how do we extend this pipeline? We've sort of shown this in pancreas cancer. Does this approach of comparing in vivo to ex vivo model systems, tuning models, how do we begin to better understand what features we need to preserve? How do we engineer models to better represent cancer cell phenotypes? And then how do we use this in a variety of sort of functional studies? And so I just want to highlight through our acknowledgements, our funding sources, a number of important collaborators through Dana-Farber, Microsoft Research, the Broad Institute, and the Farber, and of course, patients and families who have sort of supported this research. With that, happy to take any other questions or discuss more generally about some of these concepts.
Shri, just getting back to your recommendations for all of us for a framework to think about, perturb, and define cell states across many models and tissues. Sanam's kind of asking a similar question that I have about, if you imagine a dense cube of, for every tumor, there's locked in genomic contexts. There's a large universe of ex vivo growth factors and media states and dimensionalities and things you could modify. And then as a result, X times Y, molecular features of the tumor times ex vivo states produce Z, which is, I would imagine, a compendium of cell states, which then results in the drug response. So are X and Y infinite, or are there a finite PCA space of X and Y, and we should just adopt them as a community? There's 2,700 molecularly defined states in pancreas cancer. There are we could assess every growth factor and every transcription factor, but when it really comes down to it, there are 7 major cell states, and we all just need to agree to assess those 7 states in every system that we have. Is it that simple, or because I think if we can't define it, everyone's just going to wave their hands for a long time.
That's fair. Yeah,
maybe that's unfair.
No. I think this is this is a great question. I think this is sorry, I didn't see it in the chat there, but so one of the questions we ask all the time of ourselves is how much of this hypercube do you have to sample, exactly to your point, to be able to say, okay, we've covered enough? And I think I think we're trying to answer that question. I'm not sure we'll get to that answer directly necessarily through this approach. I think one of the things that we're trying to sort of work on with Microsoft Research is like, how do we sort of build this lab in the loop effort where the modeling maybe informs additional, gaps or focal points where you want to test in this space to sort of fill out and hopefully refine sort of the exact data that the right types of data that you need in this iterative process. I think the other sort of aspect to this that we're trying to build from a tool standpoint, as many of you probably know, there's the sort of PRISM pooled cell line assays that have been pooled cell lines that have been developed at the Broad. We're building a pancreas model pooled organoid, an organoid pool barcoded in the same way, and then are optimizing some methods to both do viability readouts with those, but also single cell transcriptional readouts at scale, where then maybe through these sort of high-plex tools, you can also begin to search this space in a little bit more efficiently, with selected microenvironments, cytokines, et cetera. And we've piloted some work, not published yet, and I didn't show it here, but where you can actually transduce the entire pool with a given transcription factor. So now across a variety of genetic backgrounds, can you now apply a state pressure plus a therapeutic or other insult and begin to sort of map it out? So again, I think, as you said, it's a very large search space. How do we build the tools to expand this, to sort of build out key nodes here? And then how do we leverage the computational advances also to sort of inform which pieces of this do we test? Where's our biggest bang for the buck? Really, right, in terms of testing to sort of hopefully build out, sort of identify the rules essentially governing some of this plasticity. So definitely a great question. And I don't have the answer to that, but that's sort of what we're trying to work toward. Sorry, another question. Have you seen any evidence of codependent survival advantage in the pooled sorry, for example, TF1 cells have improved survival when, oh, I see. Yeah, that's a good question. We have not tested that yet. So most of our validation, so I think the question is really, if we do TF1 and TF2 cold cultures, is there crosstalk that supports? I think this is an interesting concept, and I think we have not tested that. All of our validation has sort of been in one TF mode. And I think, one of the questions, and historically one of the reasons that sometimes pooled screens, you find it hard to see exogenous factor scores, because in a pooled setting, you can't, there's many ways to compensate essentially in a pool. So we haven't tested this yet, but definitely an interesting idea. And I think as Jesse pointed out, there's infinite number of combinations, and I think it's just like tricky for us to figure out how do you narrow that search space into sort of areas where you can learn some of the rules and then build out.
Yeah.
Yeah.
And I think, Sri, we need to end, but this, in the spirit of the SFPM, trying to define standards, metadata standards, information standards, minimal information about a FPM experiment standards, maybe you and others can guide us about how do we define growth factors, media, states with an ontology that perhaps we can all adopt, and then compare and contrast people's results against each other, as opposed to all talking about different stuff forever.
Yes. Yeah, that's right. No, I think part of our motivation, like everyone does culture to resistance, but even that, it's like, how much of that is systematic or not? And I think, again, we're trying to figure out how do you build out that space.
Shri, I think we have to wrap up, but really want to
Sure.
appreciate your thoughts, and it's an ambitious goal to be systematic about states, and I think we all share it. So thank you for leading us down this path.
Yeah. Thank you. Thanks for the invite.
All right, see you guys later. See you next month. All right,
Bye.
bye.